AP Bio Comp Book, experiment, tape to the top, Q, H, IV, DV, CV, CG, levels of treatment, replication. And don't worry about that yet. Okay, take your sheet protector, take your little experiment page, slide your little experiment page in the sheet protector, not all the way down, but just so that we can use the sheet protector to mark on before we make anything permanent. So I've got this right here, sort of at the top, but I can still see all of my stuff on my page. And so that's where the expos are going to come in. Oh, and I did forget to, I'll get you a paper towel in a minute. So we can write with the expos, but it's not going to make anything permanent onto our notebook until the point at which we want to make it permanent. Does that make sense? Okay. All right, so I want you to go ahead and take a minute and read over the experiment. Okay, remember we're not marking anything on the paper yet. We're not marking anything on the notebook. We're just going to mark on the sheet protector in the spot where it's going to be appropriate. And you're not worried about this just yet. I'll talk about that in a minute. All right, so first I have a Q here. Anybody have any idea what do you think that the Q stands for? Say that again. Quantity is going to be important, but not at this point in the experiment. This is going to stand for question. Most of the time in scientific method, you start off an experiment with a question. Sometimes, especially at the level that we're at, it doesn't specifically give you a question, but you can probably figure out what question do you think the researcher was asking or, came to mind before they came up with this experiment. So on your notebook page, write, and you don't have to start with the word what. I just put what down there. Write your question along those lines. What do you think the question is that the researcher was asking before he designed this experiment? The paper towel that I gave you, is to use to erase if you need to erase. So you don't have to start with what. That was just an example. And the Black Expo is a fine tip, so it might be easier to write with. We'll highlight things with the colored one. So, you can do whatever you want, but the black one is a little bit more fine tip, so it might be a little bit easier. And if you need beakers or anything like that, they're in the drawers. You know where all that stuff's at. Okay, so kind of wandering around, I see a lot of questions that are kind of like along the lines of which flower will the bees go to? Okay, that's a good start. We're going to take it up a notch. So, was the researcher concerned with the bees going to the flower because of the color of the flower? Was the researcher concerned about the bees going to the flower because of the smell of the flower? Why did the researcher, what was the attraction of the bees to the flower? So sugar concentration should be part of our question somewhere, because that's the attractive thing that he wanted to measure. So take a look at your question. And you don't rewrite mine. Okay, so don't rewrite mine, but this is essentially kind of summing up what I've seen on a whole lot of papers. So how can we talk about the sugar concentration? What do you think, looking at that experiment, what do you think that he predicted that the bees would prefer? Okay, so we need to add sugar concentration to it. And based on what we read in this little summary right here, it looks like he thinks he's expecting bees to prefer a lower sugar concentration. Will bees prefer flowers with, and then you can say a lower concentration, a higher concentration. You can use the actual percentages. You can use a range, 25 to 30%. So as you're adjusting yours, you can use the paper towel to wipe it off. But let's say, if I just did this, then I could mark that out and say with, and just add that information. Let's say I said, will flowers, and I'm just going to mark that out and put bees. So you can just edit it. You don't need to necessarily rewrite the whole thing. Just give yourself enough information so that when we take this off and we're writing it on this paper, that you know what you wanted to say. So we said that there was definitely a question about what the bees prefer, but then when we came back around and looked at it a little bit closer, the scientist is definitely testing preference linked to sugar concentration. Not only that, but as we read through that experiment, it looks like he thinks that a lower sugar concentration is going to show more of a preference. All right, next. What do you think the H stands for? Okay, so that's going to stand for hypothesis. So we talked a little bit about a hypothesis. Are hypotheses right or wrong? No, we never use those words right and wrong with a hypothesis. Are they correct? Incorrect? We don't use those words with a hypothesis. Chances are up to now you might have used the words supported and rejected. At this level, we're going to use the words a hypothesis is either rejected or the data failed to reject the hypothesis. Those are the ways that we're going to classify it. So failed to reject is kind of like what we used supported for in the past, but we don't use that word anymore. Technically, they're never supported. We just test and test and keep testing. Oh, failed to reject that, failed to reject that, failed to reject that, and we keep testing. We talked about, we did our little puzzle activity and how we keep an open mind about things, and when we're looking at stuff, we want to keep an open mind, but also we don't want to be wishy-washy about it. Well, it might be this or it might be that. You take might out of it. This is what it is. So when we did the little puzzle activity, we said like cows in a field. This is a picture of cows in the field, yeah? This is a picture of a galaxy, yeah? This is a picture of space. Was there anything different than that? I'm forgetting now. It's been so long ago. Oh, picture of a forest scene. Okay? So we're not going to be wishy-washy about it. This is what we're going to do. This is what it is. Because it's never right or wrong. It's either rejected or it's failed to reject. All right. In real life, we hardly ever really get that accumulating moment at the very end where we do get to take the cover off and see what the puzzle picture actually is. In real life, sometimes you go your entire life and you're like, well, this is what I think. This is what I think. And you're just gathering data to support, to fail to reject your hypothesis. Luckily for us, we have that accumulating moment. So come over here real quick. So we have all of your bits and pieces put together, and it is a scene. And we can say it is a blank. So it is a circus, or it is a, hey, oops, it is a this, or it is a that. Okay, so galaxy, forest scene, cow in a field, outer space. Anything else? Any final hypotheses? It's Friday. It's been a long week. Just don't care right now, do you? Just show us what the picture is, right? Okay, I don't blame you. Okay. Now, granted, we're missing some data because we didn't have an extra group there. But this picture, this place has a name. What's the name for this place? Where people and aliens are said to meet together. Huh? Oh, this is older than that. Any guesses? Oh my goodness, are you all that young? Have you not heard? Yes. There's an actual name for this place where people go to see aliens. There you go. So this scene was Area 51. Did anybody say that in their hypothesis? No. Does that mean you are all wrong? No. You had reasonable hypotheses, especially, you know, when we're talking about a cow in a field. If that's all you got, well, then that's what I think it is, and that's reasonable. The more data that we add to it, the more we can broaden it out and say, well, now it can't be a galaxy because there's a big van here, or it can't be a galaxy because there's a cow or something like that. So you add that and you kind of expand and keep an open mind about it. Make sense? Okay, so let's go back. Now, the good thing about this little experiment that you have is that the researcher actually does state a hypothesis. So a hypothesis is an explanation of what you think will happen. So find what the researcher, he or she, thinks is going to happen. Make sure they're explaining it. That's the hypothesis. So the researcher does give the hypothesis. Write that down on the H line. A hypothesis is an explanation of what you think is going to happen. I think that the puzzle picture is a circus because there is a bear on a unicycle and a dog in a tutu and a guy with a chair and a whip. So look for a statement like that in that little experiment and write it on hypothesis. Once again, you're working too hard. This one we can just copy. So what sentence in there looks like him making a prediction and explaining that prediction? Or her. I mean, I don't know if it's a him or a her. So we don't care what anybody thinks. No. You can look for what he thinks is going to happen, but in our hypothesis, science doesn't care. So we don't waste time with that. We start with this. So see how taking that off, it takes out the bias because it's not like I like this or I think this. Science doesn't care. Science wants to know what is the hypothesis. Oh, you took out, I think, huh? And so we just start right there. Yep. And so we don't care. We just do this. Start the sentence right there. See how it's basically the same sentence? So in science, we don't care what anybody thinks. Start right here. See how that's the same sentence. Science doesn't care what you think. Science doesn't care what you like. But if I take out that word that I think this and just start with bees will prefer, or more bees will prefer, or if the flowers have lower sugar concentration, if I just start there, it's the exact same thing, and we're taking out those words that are basically sound biased. Make sense? Okay, so I think most everybody had it on here. Something along the lines of, and of course, the researcher says significantly more bees will pollinate flower number 1, more. And that word significantly is important, but we'll talk about that when we actually get to experiments. Because if I say more bees will pollinate flower number 1 rather than 2 or 3 because lower sugar concentration. Okay, so we have to watch out for that. Because, so let's say flower number 1 had 10 bees pollinated. Flower number 2 had 9 bees. That's more bees, but does that really show that they prefer lower sugar concentration? Okay, so what if it's 11 bees and 9 bees? Is that showing that they prefer it? I mean, so where are we at? And we'll talk about those details a little bit later, but kind of keep that in the back of your mind. So the fact that they say significantly, that's still kind of a vague word. What's significant? Is 11 and 9 significant? We're going to have to be specific. So 50% more bees or twice as many bees. Something that we can collect as data and specifically say, yes, that is more. Because if it's just one more, what is that? And I neglected to say, so if you don't know what these mean, you might want to write underneath that Q stands for question, H stands for hypothesis, but if you know, then you're good. What do you think IV stands for? Okay, so this is our independent variable. The independent variable in an experiment is the one thing that's purposely changed. We only want to change one thing on purpose, right? Take one of your colored markers and use the colored marker to highlight in the experiment what you think the independent variable is. Just like underline it, go over it. Highlight what you think the independent variable is with one of your colored expos. And so in the experiment, use that and highlight where in the experiment it shows. I'm sorry, too old. I should just put my reading glasses on, that will help. Okay, now we're going to do that the same though for all of them. Now it's going to be in different corners, but they're all going to be in the pollination zone. So there is something that's a little bit better. Okay, yeah, that's keeping it nice and simple and easy, isn't it? All right, so we're putting all of them in that place right here. So since we're putting all of them in the same place, granted it's different corners, but they're all in the same place, that's something we're keeping the same, not something that we're purposely changing. There's something that's a little bit better. I don't know, is it? What do you think? Yes? Say it again. What's your question? No, just a bottle at each station. Okay, so since, here's a word right there. So there's something that's a little bit better. All right, so one of the things to kind of keep in mind when you're trying to determine the independent variable is look back to the question in the hypothesis. So that's going to be the thing that they change. So will bees prefer flowers with lower sugar concentration? If that's what I'm asking a question about, well, heck yeah, that's what I'm going to want to change, right? And so you guys did it different ways, and that's just fine. Whatever you highlighted, that's what you would write here. Now, I put one thing purposely changed. That's the definition. You should have that definition in your vocab number 1, right? So you may or may not need this here. That's up to you to decide. But whatever you highlighted up there, that's what's going to go in here right here. But you don't need to rewrite it right now. We're not going to waste that kind of time. So some people said, just highlighted this right there. That's fine. Some people highlighted this all right here. That's fine. So for independent variable, just mark it blue for just a minute because that's all we need. Take your different color highlighter. No. Yes. Never mind. Sorry. It's Friday. Long day. We're going to skip DV for just a minute, but do you know what it stands for? Okay. Let's do CV because I want to use a color on that one. So CV, use your other color expo marker. Do you know what CV stands for? Okay, controlled variables. These are things that we need to keep the same in an experiment. We need to control that potential variable. So go back through that experiment, use your second color expo marker, and highlight, underline what you think would be controlled variables. There is usually more than one. There are usually multiple variables that we try to control. Try to find as many as possible. And I'm going to give you a clue. I came up with 1, 2, 3, 4, 5, 6, 7. I came up with 7 controlled variables. Colin, did you grab those yet? Okay, you already had them. I just wanted to make sure. I came up with 7. See if you can come up with, you should come up with multiple independent variables though. I'm going to give you just about another minute and then we'll see what you got. finished or not, I'm going to go ahead and have you pause right there for just a second. So here's the thing about identifying controlled variables. For controlled variables, we can kind of bullet it. We can item list, have a list like that. It can be complete sentences. Here's the thing. It has to be specific enough so that we know what you're talking about and not so vague that it can incorporate too many different things. So like thinking back to the lab safety equipment test on the very back where it's talking about lab equipment, if you put down digital scale and then you highlighted the entire sentence that said measure 5 grams of powder and mix well in the solution, well, the measure 5 grams applies to the digital scale, but the mix well in the solution does not. And that would be something that would be marked wrong. So make sure that it's enough information that we understand, but not so super vague that it incorporates everything. Got it? The t-shirts, the hostess shirts that are on top go to Mrs. Newman, but other than that, that's all I have for you guys. Thank you so much. All right, so can somebody give me what did you say was a controlled variable? Be brave. Okay, so according to the experiment, they were all put in the pollination zone. So that's what I'm talking about. See the difference there? And I don't know what you wrote down or what you highlighted, and we didn't write anything on the notebook. But I want to point out, so if I just say the pollination zone, it's like, well, what about it? It's not a lot of information. But if we say all flowers were put in the pollination zone, that's the thing that we kept the same. Do you see why I want you to write the second one rather than the first one? Does that make sense? Okay, so pollination zone. All plants were placed in pollination zone. I got that one. What else? So you can't just write sunlight. Well, what about the sunlight? Have we filtered the sunlight? Are they getting the same amount of sunlight? So all the flowers got the same amount of sunlight. What else? Give me another controlled variable. Okay, so we can't just write water. We can say each morning they get 1 liter of water, or we can just say all the flowers receive equal amounts of water. But you can't just say water. Is it the temperature of the water? Is it the volume of the water? You know, what is it about the water? So all plants get the same volume of water. What else? So that's really weird. They're going around picking flowers off. But think about it for just a second. I have a plant that has lower concentration and a plant that has higher concentration. The plant that has higher concentration has 10 blooms. The plant that has lower concentration has 3. I count more bees at the higher concentration plant. Is that because of the concentration of sugar? We don't know. It might be, but it might simply be that there's more blooms visible, so more bees can visit it. So keeping the number of blooms the same is a controlled variable. What else? So maybe this plant over here, we do it from 1 a.m. to midnight or whatever. And this one we do just from 6 a.m. to 8 p.m. Is that fair? Is it fair to have different times? So that's a controlled variable. We monitored them for the same amount of time. What else? Come on, you can do it. All right. So can you take that? Can you kind of condense it for me? What are you saying is a controlled variable? Because actually there's like 2 different things, but okay. So that's the easy one out of there. So it's always the same number of flower in each planter. Would it be fair if this planter had 1 and that had 4 flowers in it? Wouldn't be fair. So what about the planter itself? What about the container? Would we want to keep that the same? Want the same style of planter? The same size? So absolutely. So we could say that as well. Same number of flowers in each pot. The pots are identical. So saying each planter, I think is what the word they use. Identical planters. What if I had a planter over here that's red and one over here that's black? The red one gets more bees. Might the color of the planter have something to do with it? I don't know. But we need to control as many variables as humanly possible. And so taking out the difference in color is something that we control. I didn't even have that on here. There was one other that I had. We put 3 plants in each pot, and what about those plants? They're the same type of plant in each pot. So 3 of the same type of flowers, so same number of flowers, same type of flower in each pot. Did anybody else get it? I mean, we already hit one that I didn't get on there. Did anybody else have anything that we didn't talk about? Okay, so you have multiple controlled variables. We want to control as many variables as humanly possible. All right. Next. Oops, I wrote over it. Oh, no, I didn't. CG. Any idea what that means? Control group. Have you heard of control group before? Is that on vocab number 1? Can't remember. We're going to talk about control group a little bit more later on. But in essence, your control group this is a group that doesn't get an independent variable. What's our independent variable? Sugar concentration. Is it possible to have flowers that don't have any sugar in them whatsoever? I don't know. I don't know enough about nectar to say that. What about if we use fake flowers? Are they going to have sugar in them? But it might be different, you know, if they don't have water or they don't have something for the bees to get. That might be, but that's a possibility. So sometimes you are not going to have a control group in an experiment because there's no way to do a no independent variable situation. Sometimes the question might say, okay, there is no control group, and so suggest one. How can we set it up so that there is a control group? And so that might be like, okay, well, let's have fake flowers there. Then there's a flower that has no sugar concentration in it whatsoever. All right, levels of treatment. Does anybody remember from vocab what levels of treatment were? It's Friday. We don't care anymore, right? Well, just as long as you care when you get back on Monday. It's simple. Levels of treatment. What kind of levels is your treatment? That's a controlled variable. We're keeping that the same. So we have different levels of treatment. What are our different levels? What are the different things that being treated in the experiment? Say it again. What? Oh, no. You're thinking too outside the box. So within the experiment, we're having different types of treatment. We're changing how we do things. So the levels of treatment are linked to our independent variable because the independent variable is what we're purposely changing. So how are we treating things differently? How are we having different treatments? Not necessarily get, but have. Okay, so how many different levels of treatment do we have? We have 3 different levels of treatment. 17%, 54, and 71. So we have 3 levels of treatment. 17%, 54%, and 70 what? 71% sugar concentration. That's our levels of treatment. That's how we're having different treatments within that experiment. Okay, last but not least, replication. If I replicate something, what am I doing? Okay, so replication, you're repeating, you're doing it over and over again, you're making a copy. So in this experiment, did they just do it once? I'll get that in a minute.
For today's activity to
All of
report
them.
to the break area by the vending machines in the front entrance. All other students need to be standing outside between the 2 buildings in the back, between the main building and the building that has construction and HVAC. Thank you.
Okay, so the instructions that I got yesterday is we do this during break time, but apparently we're doing this early. I'm going to hang on to Gabby, so I'm sorry. I'm going to kick you out of the little activity that they're doing just so that we can go over and get a few other things caught up. The rest of you guys, they have a little activity. It's kind of a little search. I think they call it the Amazing Race. So you're kind of going around campus. We need a person to be the team leader, and that's fine. Do you have QR code scanning on your phone? Okay, so y'all agree? Okay, so that just makes it easy. All right, so Kylie, I'm going to email you right now a link, and you need to click on the link, okay?